For as long as humans have been aware of themselves, they have noticed that children look like their parents. And of course, they don't look exactly like their parents. They're certainly not clones. And there's a variability between different kids in the same family and how much each of them look like their parents. This inheritance has been something that people have been curious about. And by 19th century, so this is when Charles Darwin is around, the prevailing idea in Western Europe was that it was the mixing of blood during conception that caused maternal and paternal traits to mix up, and the child then therefore is some sort of a hybrid of the two parents. This is where we get phrases like pure blood and royal blood. Charles Darwin really struggled with this concept because remember that in his proposal of evolution by natural selection, individuals in a population have diversity among them. And some of those individuals survive and some do not. The ones who survive are the ones who have a trait that allow them to get more resources or survive against predators or disease in whatever way. Something about their trait allowed them to survive better, reproduce, and pass the same traits that allowed them to survive to the next generation. But how did those traits get passed exactly, like physically? How did that information from parent to offspring get moved around? Darwin had a hypothesis, and it has not been supported by any science. It was rejected pretty early on. This would be a frustrating mystery to Darwin for the rest of his life. And the irony of that is that he actually had the answer to his question right in his own home office. Dun dun dun. I'll tell you more about that at the end. So in this lecture, we will learn about the father of genetics, Gregor Mendel. We'll learn about his monohybrid crosses, test crosses, and dihybrid crosses. We'll see how the basis of inheritance is actually in chromosomes and not blood. And then talk about some traits that are more complex, and we call them extensions to Mendel because they follow the same concept as Mendel's laws, but there's more complexity built on top of them. We'll look at X-linked traits and some other human traits that are of interest. Gregor Mendel was born, actually Johannes Mendel, in a German-speaking family, but on the border of Moravia and Silesia, which is modern-day Czech Republic. I think his mom was actually of Slavic origin, so she was probably Czech. And his dad was German-speaking because that part of Europe, as most of Central and Eastern, well, I shouldn't say Eastern, Central Europe was under the Austrian Empire. So he was a son of a farmer. They had a farm for the 130 years before his birth. I think he had two or three sisters, and was just a nice guy. I like reading about him because he just has such a humbling, interesting story. Well, he decided to enter the monastery in the local town called Brno in eastern Czechia, Czech Republic. And the reason for that was, as crazy as it sounds now, monasteries were some of the most progressive education institutions of their time. So there were people who did research, philosophy, communicated with other people. And Mendel himself even said that he was choosing it because he's not going to have to worry about having food to eat and putting food on the table for his kids, because he's not going to have kids as a monk, and he's going to be part of this. I mean, monasteries are kind of like communes, right? They kind of work together and take care of each other, and he wouldn't have all the like daily worries. He wanted to also become a teacher because, yes, you can be a monk and a teacher. And his dream was crushed when he failed the oral examination part of the teaching education. And then he ended up taking more schooling at a university, which was sponsored by the monastery that he was now a part of. And at the end of that second round of education, he went through his exams to become a full credited teacher again, and unfortunately once again failed the oral examination and never did become a teacher. So it was a huge bummer, big disappointment for him. But as we will see, he ended up having a very huge positive impact on the world in another way. Since he was a farmer and had interest in, hybridizing, breeding different kind of plants on his farm where his family had lived, he was also interested in this question of how do traits get inherited by the new generation? How do parents pass on their traits to their offspring? And he decided to do some experiments just to grow a few plants in his garden in the monastery and ask some of these questions, collect data, and see what he gets. He decided to work with a humble pea plant because peas are really easy to grow in this region in Central Europe. There are many varieties that have distinct features, and you can breed several generations in one growing year. Oops, I don't know what I was thinking there. And this is the monastery that he was a member of and where he had his garden, including the pea garden. I took this photo last year, actually, or has it been 2 years now? I really like Mendel's story, and it was exciting for me to be able to visit Brno, and there are several genetics museums that are really cool, engaging, as well as a small museum inside of this monastery that honors Mendel and some of his work. And of course, I met Mendel, or the wax statue of him. Don't worry, I wasn't actually touching him, and it was totally okay. Pre-COVID, so Mendel nor I are actually wearing masks because we're unaware of all the respiratory viruses that are coming our way. So let's talk about the first thing that he worked on. Now, in retrospect, we call this set of experiments monohybrid crosses. What this means is there's only one trait that he's going to be keeping track of. Okay? I want to show you a little bit of how he did it. So this at the top here is a flower of the pea plant. Most flowers have both male and female parts all on one flower. This is not true for all flowers, but it is true for pea plants. So this part in the middle that's a little bit hard to see in this picture is the female part, and the stamen here are like little popsicles on a really thin stick with a bunch of pollen all over are the male parts. And the way that it works is the wind blows the pollen, it sticks here on the female stigma, and then the pollen actually grows a tube that travels all the way down into the carpel, which is kind of like the ovary of the flower. And then the pollen, which has a sperm cell inside of it, fertilizes the egg or eggs that are inside there. Then the petals of the flower and the anthers, the male parts, they fall off, while the style and the ovary here, they develop into fruit. So when you're eating fruit of a plant, which means many of the vegetables are fruits like tomatoes and cucumbers, you are actually eating the developed ovary that has fertilized eggs in it, right, which become seeds. Then you take each of these fertilized eggs, which have now become seeds, and you can plant them, and from that, an entire new plant will grow, right? Okay, so he was able to manipulate some of this natural process by crossing different plants with one another. So what he would do is he would cut off the anthers, the male parts, off of these purple flowers, for example. Then he would use a paintbrush to dab the pollen from a different plant, and then he would tap it onto the female part of that plant that doesn't have any pollen on it. So in this way, you can ensure that it is the sperm from the white flowered plant that fertilizes the eggs of the purple flower plant. So this was his way to control the mating instead of leaving it to wind or random pollinators like bees that may come and get pollen from all over, from all kinds of different plants, and then come and possibly fertilize one of these flowers. Okay, so this is the technique that is used. You basically artificially fertilize and select who gets to mate with whom. His goal was to study two variations of a single trait. So in today's language, we would say two variations of a single gene. And he started this process by producing or ensuring that he had true breeding pea plants for 7 different traits. Okay, what does true breeding means? It means that if you have a plant that when they are able to self-fertilize, meaning the sperm and the egg comes from the same individual, then their offspring looks exactly like the parent for that trait. So let's say here, if this purple flowered pea plant was able to fertilize itself, and all of the offspring from that fertilization, once they were planted and grew, also developed purple flowers, and then you take these second generation, you make sure that they fertilize themselves only. So each individual plant fertilizes itself, and then you get seeds. When you plant them, they only grow to produce purple flowers. You would say that this plant is true breeding for purple flowers. So we're talking about one trait that if you allow the plant to self-fertilize, it will continue to have that exact same trait as the parent over and over and over for unlimited generations. He did this for 7 different traits. Flower color, so he found purple true breeding plants. He found separately white true breeding pea plants, meaning white flowered. Ones that had flower position axial or only terminal, so flowers grow at the top of the stem versus on the sides. Seed color, yellow-green. Seed shape, round or wrinkled. Pod shape, pod color, and stem length. Okay, so he had 14, you could say, containers of seeds. And each of those containers of seeds were true breeding pea plants for a particular trait, right? True breeding for purple flower, true breeding for white flower, true breeding for yellow seed, true breeding for green seed, and so on for all of 14 of these variations of the traits. Okay. So now that you know how he was all set up, let's look at the experiment that he did, his first set of experiments. He grabbed that true breeding purple flowered pea seeds and he planted them, and they all grew to be mature plants with purple flowers. Also, in his garden, he planted the true breeding white flower pea plants, put them in the ground, watered them, watched them, and they all grew to have white flowers only. Okay, so he has mature plants that are true breeding purple flower and true breeding white flower. Then he very carefully did that whole artificial breeding technique. We say that he crossed or artificially pollinated them with one another. So made sure that purple was the mom and, so to speak, for plants, and the white was dad, or opposite. He did both ways. Then those flower petals fell off and the female part of the flower developed into a pea pod, and he saved those seeds and then he planted those seeds. So the seeds from this cross were planted in the ground. When they started growing and they started maturing, he noticed that 100% all of the offspring of that cross produced purpled flower. They were purple flowered. They weren't like light purple or purple with white dots or some sort of an in-between color, diluted or pale. They were the exact purple that the purple parent looked. So this was the interesting question. Like, look, I just crossed these two different flower-colored plants, but somehow I'm not seeing any white flowers in this offspring generation. He called that first offspring generation of the cross F1 generation. It stood for first filial generation. The word filial is not used very much, but it means of or due from a son or daughter. Okay, so it is related. He didn't just make up some random word. Okay, so mystery, like, you crossed a purple parent with a white parent and all the offspring are 100% purple. What he did next is very, very important. He allowed all of these F1 generation plants to self-pollinate. So they were, this one individual plant is both the male and the female, or the mom and the dad. Then when he allowed them to self-pollinate, he collected the seeds from that fertilization and planted those seeds and waited for them to grow. And they grew into mature plants, which started to flower. What he saw in the result, which he called the F2 generation, is that there were now mostly purple flowered plants, but also some white flowered plants. This again was really interesting and kind of shocking, because imagine that for all of these plants, Both parents, mom and dad, were purple looking. They were purple flowered plants. And yet the offspring, some of them are white flowered plants. How is that possible? Where did the whiteness for this flower color come from? So the offspring resulting from this self-fertilization of F1 plants is what we call the F2 generation. And although it was hidden in the first filial generation, there is a trait that whiteness reappeared in the F2 individuals. And when he counted how many there were, which there were thousands of plants that he planted, it was about a 3-to-1 ratio of purple to white flowered pea plants in this F2 generation. And you need to remember this ratio of the F2 generation of a monohybrid cross is 3-to-1. He called, or rather, actually I can't remember if he called it or if we're calling it now. He called the trait that was the only trait of this setup in the F1 generation. So there was only purpleness. He called that trait dominant. And the one that happened to be hidden in the F1 generation, he called that one recessive. Okay, so when you breed two varieties of some trait that are both true breeding, the offspring of that cross will only look like one parent. Whichever parent it happens to look like, that trait, in this case, this purple coloredness, is dominant. And the one that happens to be hidden in that F1 generation is by definition the recessive variant of the trait. Okay, he did this many, many times over several years. And he didn't only use his true breeding seeds for flower color, he also did the same type of experiment and collected data from all these other pairings of variants of traits. And notice how many individual plants he was counting. I mean, you have 1,000 plants here for this flower color, and then when we're talking about the seed color, it's many thousands of plants. And in every case, the ratio of the dominant to recessive trait that pops back up in that F2 generation is 3-to-1. So there are 75% of the dominant trait that come up in this F2 generation, and 25% of all individuals in the F2 generation are white. This is all these percentages and all of this is very, very specific to this exact setup that he had. So you had to start out with two types of true breeding plants for the same trait. So the trait is flower color, one variant is purple, one is white, and they're both true breeding for these variants. You artificially cross-fertilize these two true breeding plants, and you have the F1 generation where 100% of the offspring come out to look like only one of the parents for the trait that you're keeping track of. So he doesn't care about any of the other traits in this experiment. He's only looking at flower color. Okay. And then when he lets F1 self-fertilize or self-pollinate, the offspring, 75% of them are the same color as F1, while 25% of them are now revealing the trait of that recessive variant. So I really encourage you at this point to pause this presentation and to retell the story of Mendel's monohybrid crosses as if you were explaining it to a classmate who missed this lecture. So start with Mendel was doing plant experiments with peas. This is what he got to get started. Make sure you understand what it means when we say true breeding plants, and explain that to your imaginary classmate that you're talking to. And then tell him what he did. So here he did 2 rounds, 2 generations before he got to the result that yielded the 3-to-1 ratio. And then go ahead and come back after you've told the story. If there's any confusion, please consult your textbook or go back and see if it'd be helpful to listen to some of this again. I hope you're really solid on the story of the monohybrid cross in Mendel's pea garden up until this point, because it gets more confusing if you are not quite solid with this material. So as he began to analyze the F2 generation and why it has this 3-to-1 ratio, he was kind of limited by the science or the technology or both that he had access to at the time. But he did figure out that the ratio actually was a surface ratio, and that deeper on the inside, the ratio was actually 1 to 2 to 1. That's super mega confusing. So let me show you what that means. He discovered that out of all of the plants that he got for the F2 generation, 1/4 of them were true breeding dominant. 50% of them were not true breeding, and 25% of all the F2 generation plants were true breeding recessive plant. This is what that means. Remember that the original parent that he started with, which we called the P generation, he had a true breeding purple pea plant, which means that inside of this plant, there is only information to ever make offspring that are purple colored. That's why when you allow this plant to self-fertilize, it will produce purple plants. And if you let it self-fertilize and make sure it only is using its own pollen to fertilize its own eggs, it will always produce flowers that are purple, flowers that are purple. And remember that the other parent that he used was a true breeding white pea plant. That means that in the cells, which he did not know much about that at that point, there's no concept of DNA at this point, but he knew that whatever it was, the information for flower color in this parent was only to make white colored pea plants, because anytime you allow this plant to self-fertilize, it only produces white flowered plants indefinitely. However, when you cross-fertilize these 2 different types of true breeding pea plants, all of the offspring are not, all the offspring are purple flowering offspring. However, this generation of purple flowered peas are not true breeding, because when he let this F1 generation, after the first cross, when he let them self-fertilize, some of the offspring, remember plants can produce hundreds of offspring because of all the seeds, right? Some of the offspring were white. By definition, true breeding, all of the offspring needed to have that same exact trait that the parent had. And here you go. It's proving that not 100% of them did. And when he allowed these F2 generation plants self-fertilize to see, well, I'm going to let this guy breed with himself and see if he's true breeding, he found out that that plant was true breeding. and that the white plants were all true breeding. But about 50% of all the plants in the F2 generation, when he let them self-fertilize, they produced both colored flowers, meaning they were not true breeding. So again, out of the F2 generation, to the naked eye, the ratio was 3-1 or 75% to 25% purple flowers to white flowered plants. And remember, for this pea plants, all the flowers on one plant are the same color. But under the surface, when he did all those follow-up experiments to figure out if they're true breeding or not, he found out that all the white ones are true breeding, but out of the purple ones, a bunch are not true breeding. And the ratios for those hidden information was 1 to 2 to 1. Okay, I recommend that you pause one more time to restate out loud to yourself or someone next to you what I have just explained about the F2 generation. And what I mean when I say 3-1 is actually 1 to 2 to 1. All right, welcome back. The conclusions that he came to is that his plants definitely did not show any kind of blending the way that people talk about blood blending as if it's like paint. You know, you have yellow and blue and then you mix it together and you get green colored something. That's not what he saw. Clearly, from that very, very, very first cross that he made, when he crossed a purple flowered plant with white flowered plant, the offspring were all completely purple. There was no blending. So he concluded that there are no intermediate traits and no blending, and that each trait is intact, discrete piece of information that gets passed on. So there's information for purpleness and information for whiteness. For each pair, so remember I showed you he looked at 7 traits and each trait had 2 variants. Well, for each trait, one of the variants was dominant and one was recessive. I think the word trait here is used in a confusing manner, so I apologize. But I hope the reason for this is because we're trying not to use the super scientific terms yet, but we will come to them in just a few slides. Okay, so in the case of the flowers for the pea plants, the purple is dominant over the white. Alternative traits were expressed in the F2 generation in the ratio 3/4 dominant to 1/4 recessive. So this is 3-1 or 75% to 25%. And he eventually described a 5-element model that we have altered just a little bit with more modern language. So parents transmit discrete factors, meaning they pass on discrete pieces of information. So like a recipe, and this is called a gene in modern terms. This word did not exist in Mendel's time. Each individual, so if you're a pea plant or a human, you receive 1 copy of a gene from each parent. And this is called the law of segregation. We're going to come back to it in a minute. Not all copies of the gene are identical. This is something that I tried to tell you when I was first explaining what a chromosome was and what a gene was. And I mentioned that you can have a gene for eye color, but there can be blue eye color versus brown eye color, right? So same gene, or quote recipe, can have variations, and the variations of a gene are called alleles. So alleles are alternative form of a single gene. In the case that we've been talking about so far, the pea flower color has two alleles, the purple allele and the white allele. If you are a true breeding pea plant for the flower color, we called you true breeding, and that's what Mendel used because that's the term that kind of all the farmers used, which is where this information was relevant at the time. But now in modern genetics, we say that true breeding, meaning you have two of the same alleles for a particular gene, we say that you are homozygous for that gene. If you have two alleles that are different, then you are heterozygous for that gene. So if I can move back for a second, this guy was homozygous for the flower color, and he was homozygous dominant. This guy, this other parent that we call true breeding, is homozygous recessive for the flower color gene. Whereas this guy here of the F1 generation, he's heterozygous. Okay, so we don't say dominant or recessive because he has both alleles. He has both varieties. They're just hidden and only one is being expressed. And again, right here, the left panel of the F2 generation is homozygous dominant. Can you pause and say what these other ones are? Okay, let's check how you did. So the left one I already said is homozygous dominant, and we only know, you wouldn't know it by looking at the plant. The only way to know it is to let it self-fertilize, breed it, and then see what all those offspring look like. And then you see, oh, it only had purpleness in it. It only had information for purpleness because all the offspring are purple. So we can conclude that this was a homozygous dominant individual. Whereas both of these examples over here are heterozygous. Again, by looking at all the purple flowered plants, you wouldn't be able to tell by just looking at them which one is homozygous for the purpleness and which ones are heterozygous. The only way to know it is to do all these breeding experiments. This white plant right here is homozygous recessive. It only has information for the whiteness, and we know that the whiteness is recessive to the purple. How do we know that? Because in a situation when both of those traits are combined in a single individual, right? The seed that grew into this plant had both information about purpleness and information about white, but for whatever reason, it only made the purpleness for the flowers. And just by that, we define purple as dominant. So if there's both traits involved, but only one of them is expressed, then that trait that's expressed is called dominant over the other one. That fourth point was that alleles remain discrete and they did not blend. There was nothing light purple or purple and white stripes or blotches or polka dots or anything like that. They remained totally discrete, unblended. The presence of allele does not guarantee expression. Remember that F1 generation, they were all purple, but they did contain in them information for how to make whiteness, and that ended up popping up in the future generations, right? So just because someone looks to have a certain trait, like you look purple, it doesn't mean that there's only purpleness information inside. Right? And two other terms here, genotype is the actual alleles that an individual contains. So in the F1 generation, that first offspring generation, that set of plants had purple and whiteness inside, so we call that genotype. Genotype cannot be seen with the naked eye. Genotype can only be found by experimentation or some other invasive, deep scientific procedures. Phenotype is the actual physical appearance, so that's something that you can tell by looking at it. So phenotype is purpleness, or phenotype is the whiteness in this particular example. Okay, so when someone says, what is the phenotype of that individual, they're usually talking about like, what trait can you see or measure with kind of your naked eye without looking at what's inside the cells. As promised, we're coming back to the principle of segregation. The official definition of the principle of segregation is two alleles, remember that's versions, of a gene, which is like the recipe, segregate during gamete formation and are rejoined at random, one from each parent, during fertilization. It's almost like you have to translate that sentence. So I encourage you to pause here and see if you know all the terms already. Go ahead and see if you can digest what that very dense sentence is saying. All right, I hope you did that pausing and digesting. I will show you on a diagram what this means, and it all has to do with what happens during meiosis, which is the formations of eggs and sperm learned about in the last chapter. And of course, Mendel at this point did not know anything about chromosomes or meiosis. None of that has been discovered or described yet at all. So look at this diagram, and remember here you learned in the previous chapter that you receive, let's say this is chromosome 1, that largest chromosome that I showed you in the fluorescent picture. You receive one of those from mom and you receive one of those from dad. And in the same area of both of those, you have information for how to make flower color. So let's zoom in on that. Remember that chromosome is made out of DNA double helix that is wrapped around histone proteins that are condensed on themselves. And it is the order of nucleotides in that section of DNA that creates the code for the trait. So just like a recipe or the fact that different order of letters written down on a page have a different meaning to you, right? Like A-N-D is different than D-A-N, even though it uses the same letters, right? It has a different meaning. And versus Dan. Okay, so notice how here the genetic code, remember nucleotides are made of ribose sugar, nitrogenous base, and a phosphate, and it's the nitrogenous base that is responsible for the code, while the other part of DNA nucleotides are identical, and they just form this kind of backbone of the DNA double helix, while the nucleotides nitrogenous bases are the ones that stick out. So see how here it's TATTTAGCCA, while from the other parent you inherited in that same area a code that goes GATTTAGCCA. Notice there's one difference here. Because of that one difference, and sometimes it's more than one, and normally genes are thousands of nucleotides long, so we're just using a smaller example. Notice that one nucleotide difference in the genetic code makes it so that when this code is read by your cellular machinery, it gets a message of make purpleness in the flowers, whereas this one reads make whiteness in the flower. In reality, in most things, especially with plants, if it's white, it just means it makes no pigment, whereas reading this code, it makes a pigment that reflects purple wavelengths, so it looks purple to our eyes. Okay, so one more time, the two homologous chromosomes you inherit from each parent, and you have 23 sets of homologous chromosomes that you inherit. Each pair of homologous chromosomes carries the same genes on them, but the types of genes or the versions of them are different. So we would say that this is the P flower color gene, and these two different codes are two alleles for the flower color, or two variations for the flower color. So alleles are versions of the same gene. And this individual we're looking at, are they homozygous for flower color or heterozygous for flower color? Okay, they're heterozygous for flower color. So if you had this in your diploid cell that then went through meiosis to create an egg or a sperm, then you know that each egg or each sperm will only receive one of these. It will either receive this chromosome or it will receive this chromosome, right? So if each egg has only one of these chromosomes, and each sperm has one of these chromosomes, then during fertilization, when egg and sperm combine to create a zygote, which is finally again a diploid cell, you will receive again one chromosome from mom and one from dad, and we'll be back to where we're started here. That is taking into account a lot of stuff that we've talked about in the past, especially a chapter on meiosis. So I encourage you to pause and go over this before moving on. You're also welcome to note down any questions that you're not able to figure out after going through it on your own. In order to kind of be able to predict statistically, oh, what if I have this many of this type of plant and I breed them with a different variety of that same plant, what will happen? Something that was put together is called a Punnett square. You may have done these if you took high school biology. I remember that I did, although I didn't really understand what was going on. And they're just, it's pretty straightforward, but there are a couple of rules that when you write a uppercase letter, that represents a dominant allele. When you write a lowercase letter, it represents the recessive allele of that same gene. So in this case, they're both P for purple, even though the lowercase p actually stands for white. Okay, so they're both P's, they're both the same letter, and you can pick whatever letter you want. But they're both the same letter because they're both representing the same gene, and the gene is the flower color gene. They're slightly different, uppercase versus lowercase, because they are representing the slight differences, which is the two different alleles. Okay, so true breeding white flowered plant is the homozygous recessive plant, and that's little p, little p. Why does it have two letters? Because the plant itself is diploid. Just like most cells in humans are diploid, they have two sets of chromosomes, one from mom, one from dad. That's why you have two alleles, and that's represented by two letters. Okay. The only cells that have one allele of each trait are gametes, eggs and sperm, because those are haploid cells. So let's look at how this works. Okay, so men on top, that's how I remember that the sperm come on top. You draw a little square and you say, hey, if I know that my male partner here is heterozygous, that means that 50% of the sperm that male makes, 50% of them will get the dominant allele, and 50% will get the other recessive allele. So one more time, when a mature adult, human or plant, go through meiosis, they begin with a diploid cell, which has 2 of each allele. When that individual makes sperm, in this case, their sperm is going to be haploid. It's only going to have one set of chromosomes, so it can only have one version of each gene. Therefore, some of them, by that, remember how meiosis kind of shuffles things around until every one of the 4 gametes at the end of meiosis gets one of each chromosome. So that means 50% of the sperm will have the dominant allele, 50% will have the recessive allele. If the female, we're looking now on the side, if the female was true breeding, or in other words, homozygous, this would look, that's not on this page, sorry. In this case, the female is also heterozygous, and therefore 50% of her eggs will have the dominant allele, 50% will have the recessive allele. So this is actually crossing that F1 generation with itself. Remember that the F1 generation were the purple flowered plants that actually had both information from the purple flower parent and from the white flower parent. So all of those individuals were heterozygous. Therefore, when they themselves produced sperm, they had half of each kind. And when those individuals produced eggs, they had 50% with the purple, 50% with the white. And then you just fill in the little table, which there are 4 spots here. It's like if this sperm that has the little p happens to fertilize this egg that has little p, then the offspring will have 2 white alleles. They'll be homozygous for the recessive allele. So you draw it all in until you have this final. And this is your completed Punnett square. What that means is that when you cross one heterozygous individual with another heterozygous individual, 25% will be homozygous dominant, 25% will be homozygous recessive, and then 50% will be heterozygous. So they'll have one of each allele. And that's exactly what happened when right here at the bottom, the F1 generation was allowed to breed with itself, with the F1 generation, heterozygous with heterozygous. This is exactly what Mendel saw. Now, moving into the past one step, one generation, when you had true breeding white crossed with true breeding purple, what you actually received is that all 100% of the offspring turned out to be heterozygous. They all looked purple, which we would say they had purple phenotype, but their genotype, what was actually in their genes, was one of each allele. Okay, so here's another summary of what happened. I encourage you to take a pause and go over that last section on your own, again, explaining it out loud to your imaginary friend and trying to figure out any of the confusion that may have come a long way by reading the book or going back over the previous parts of the video. And then when you're ready to move on, we'll talk about the test cross. A test cross is like an experimental technique in order to figure out the genotype of something. Because what are the 2 potential genotypes for individuals that have a dominant phenotype? Okay, so what that means, individual that has a dominant phenotype would be you grow peas, You don't know what you're doing, but the pea plants that grew are purpled flowered. And you're wondering, is this particular plant, and I'm touching a plant, this particular plant that looks completely purple on its flowers, I cannot tell if it's a true breeding purple plant, meaning dominant homozygous plant for purple flower color, or is this a heterozygous purple flower plant, meaning that it actually has information in it or an allele for white colored. Okay. Test cross is to help you figure out the answer to that question. If an individual looks dominant phenotype, they can be either homozygous dominant or they can be heterozygous for that particular trait or for that particular gene. Test cross is a way to distinguish between those two. Okay, so cross used to determine the genotype of an individual with a dominant phenotype. And the way you do that is you take your unknown dominant looking individual and you cross them with a homozygous recessive individual. And the reason why you can do that easily is because homozygous recessive individuals, they only have one phenotype. They all look the recessive type. In our previous example, all of the white flowered pea plants, they were true breeding for white color, meaning they were homozygous recessive. They did not have any other versions of information for the flower color. They only had the whiteness. So we take our unknown individual that looks dominant. We know that it has at least one dominant allele, but we don't know if this other one is dominant or recessive, and we cross them with a known homozygous recessive. And depending what you get, you can tell whether that individual that had the dominant phenotype is homozygous or heterozygous. This is what they would look like if in the end all of the offspring from that test cross are purple, or they all look like the dominant individual, then it means that the original mystery individual was homozygous for the trait that you were seeing. If the result of your test cross yielded individuals that looked both phenotypes, that had both of the phenotypes, then it must mean that the mystery individual that looked dominant was actually a heterozygous individual. Please pause the video and look through the Punnett squares on this page in this example and see if that makes sense. So if I were to give you a result of a Punnett square on an exam from a test cross, would you be able to tell me if the mystery individual who had the dominant appearance, if they were homozygous or heterozygous? Another set of experiments that Mendel did, we call now dihybrid crosses. Actually, he may have come up with that term. Dihybrid crosses take into account two traits. And his question is, how are the two traits passed from one generation to the next? So for example, if he had two true breeding lines with two traits, so peas that had yellow seeds, and those seeds were also round, versus pea plants that had green seeds that were also wrinkled. So here we have two traits, the color of the seed and the shape of the seed. What he was curious about is, if something is yellow and round, will it always stay yellow or round? Like, are the two traits, the shape and the color, somehow linked to one another and get passed on together to the next generation. Okay, so if we look at the genotype of both of these true breeding plants, we're talking about two sets of chromosomes, two pairs of chromosomes. One pair has the gene for seed color on it, and one pair has the seed shape on it. The yellow round true breeding plant is homozygous for both of those traits, for being yellow and for being round. The other true breeding plant is homozygous for both of those characters, green and wrinkly. When he crossed these true breeding parental types, so that the ones that I'm showing you right here, big R, big Y with little r, little y, little y. The F1 generation, all of the offspring looked like the yellow parent. They were both yellow and round, all of them, 100% of them. So he determined that yellowness is dominant over green and round is dominant over the wrinkled shape. So the phenotype is that they looked yellow and round. However, their genotype, we now know, did have the information for how to make a green and wrinkled. It's just that they were hidden or covered by the dominant traits of yellow and round. Then he allowed these F1 generation, so this phenotype, to self-fertilize and produce the F2 generation. And what he saw in the F2 generation is a ratio of phenotypes 9:3 to 3:1. In other words, he saw all different combinations of these traits. So here is the Punnett square that represents it. Now, Punnett squares for dihybrid crosses are twice as big, and it does seem more complicated to the student when he first or she first or they first encounter a dihybrid Punnett square. So let's look at what is happening here and how you would do a dihybrid Punnett square. So first you have to determine, the first step is you have to determine which allele, that's version of a gene, goes into the gamete for each trait. So we know that if there is true breeding yellow and round, meaning homozygous dominant plant, when that individual plant produces its gametes, eggs and sperm, each of those gametes will get one copy of the round trait and one copy of the yellow trait. And the reason for that is because we're not now talking about one pair of homologous chromosomes. We're talking about two pairs of homologous chromosomes. So in this diagram, let's say, again, on the chromosome number 1 for peas, there is the shape allele, the shape gene. And on a different chromosome, let's say chromosome number 4, there is the color gene, right? And in this case, the color gene is yellow, and in this case, the shape gene is round. So you make a combination of all of the gametes that are possible in the sperm up here and all the gametes that are possible in the sperm over here. This particular visual is showing us a Punnett square for the F1 cross. So remember that first filial generation is crossed. They were all round and yellow. He crossed them, he let them cross with themselves, and the products of those crosses are called the F2 generation. So if that F1 generation was heterozygous, that means that it had both big R and little r, big Y and little y inside of its nuclei, right? It had information for both varieties of both of the traits. So when that plant makes eggs and sperm, some of the eggs and sperm will get one of the round, one of the yellow alleles. Some will get combination of round and green allele. Some will get wrinkled and yellow allele, and some will get wrinkled and green allele. And since you self-pollinate, the same options are on the left. Then you just simply bring these letters over here and cross it where it intersects with these letters. And you always put the letters next to each other just to make it easier. So you don't put RY, RY. You put RR, YY. Okay. Notice that after you complete the Punnett square, which gives you the genotypes of each individual possible, then you can look at what the phenotype will be, right? Always keeping in mind that yellow is always going to overpower the green allele. So the phenotype, if you even have one yellow allele, the phenotype will be yellow, and round overpowers or is dominant over the wrinkled. So here, I encourage you to please pause the video and make some sense of this dihybrid Punnett square cross and see if you would be able to explain it to someone next to you in an imaginary scenario where you would have to do that. The principle of independent assortment is what Mendel called the way that two different traits that are observed together are passed on from generation to generation. So in a dihybrid cross, the alleles of each gene sort independently, meaning the way that the yellowness gets passed on has nothing to do with how the roundness or wrinkledness gets passed on. They are separate and they segregate separately. The segregation of the different allele pairs is independent. This is because of the independent alignment of different homologous chromosomes during metaphase 1. So remember how we said they can align at the metaphase plate. The tetrads in meiosis 1 can align at metaphase plate facing various directions, and how one tetrad aligns has nothing to do with how the other tetrad aligns, and that therefore the chromosomes can be separated separately. So as long as the two traits we're talking about, in this case, the shape and color of the seeds, as long as they are physically located on separate chromosomes, then this principle of independent assortment applies. So let's look at understanding this principle for a dihybrid cross by looking at meiosis again. So here we're looking at this parent. Inside of it, we're only going to look at 2 pairs of chromosomes because although P's, I think, have 8 pairs of chromosomes to make it easier to keep track of what's going on. So on this longer chromosome, we have this seed shape. This is the one they got from their mom, and this is the one they got from their dad. You see how this plant we would call true breeding in the plant breeding world, because it only has, as far as seed color, information for yellow, on both of its chromosomes and information for round. So as long as it keeps self-fertilizing, it would only have that information to keep passing on, right? So when it makes its gametes, all of the gametes get one copy of the chromosome with the round allele and one copy of the chromosome with the yellow allele. In this other true breeding parent that we started with, similar situation, except that they end up putting into their gamete a chromosome that's carrying the wrinkled and the chromosome that's carrying the green allele. Okay. When they have their offspring, when these two parental true breeding types or homozygous types get cross fertilized, all of the offspring look the same. They're all yellow and round, and they all have one of each chromosome, right? Because it's just a simple combination of their parental chromosomes. If you allow that plant to self-fertilize, which is like mating with someone exactly the same genetically, here is what we get. If the tetrads, and here's one tetrad, these 4 chromatids, another tetrad, these 4 chromatids, right? They're all duplicated because S phase has happened during interphase, and this is meiosis 1. The tetrad is on the metaphase plate in the middle of the cell, so we're in metaphase 1 of meiosis 1. If they happen to line up this way, you see that the 2 dominant alleles are on the left-hand side. And so meiosis 1, the homologs separate. Meiosis 2, the sisters separate. And so you get gametes that have both of the dominant types and both of the recessive types. That's all because of this alignment here. If by chance the tetrads are aligned like this, where the maternal and paternal chromosomes are not facing the same direction, then when they get split up in meiosis 1, and then again in meiosis 2, sister chromatids separate, then the gametes get one of each chromosomes, a dominant and a recessive, in its gamete. I encourage you to pause the video and slowly go through and make as much sense as possible over connecting what happens in the F2 generation of dihybrid cross in light of what happens during meiosis. It's very important to understand that. What we've talked about so far is now referred to as Mendelian genetics or classic genetics. We now know that everything that Mendel described is accurate and has been supported by decades of data from all over the world. Most important of which is that traits do not blend, but they are discrete units of information. They get passed on from parent to offspring on the physical chromosomes, and therefore they separate and segregate during meiosis. When 2 gametes, so egg and sperm, unite during fertilization, then the 2 haploid cells fuse together to make a diploid zygote cell, which then restores diploid structure to a cell, and they have 2 copies of each of the allele. However, you may look around or even look at yourself in the mirror and say, wait a minute, traits do too blend. I mean, my mom is tall and my dad is short and I'm kind of medium size, right? So let's look at how all of that still fits into and is supported by the laws and the patterns that Mendel discovered. In his model, the inheritance follows the laws as long as Each trait is controlled by a single gene. Each gene has only two alleles, so purple and white flower alleles, and there's not purple, white, and orange. And there's a clear dominant recessive relationship between the alleles, where purple flowered pea plants bred with white flower pea plants that are true breeding produce totally purple, just like the purple parent, heterozygous offspring, right? However, most genes do not meet these criteria. And we will look at some examples of what that means. However, I want you to keep in mind that this does not negate or change anything about the laws that we got from Mendel's experiments. It means that Mendel discovered the very basis of genetic inheritance, and all of these other situations that I'm going to mention, they are like extra layer of complexity on top of the basic genetic inheritance that Mendel described. It was almost like he was extremely lucky to have chosen a plant and traits that were the most basic and easiest way to demonstrate what he wanted to demonstrate. It seems like it's super, super lucky, but in reality, he actually worked with mice before he worked with peas. But the superiors at his monastery were not fans of Mendel breeding mice in the monastery and, quote, studying their sex because, they had to breed the mice in order to get the next generation. He also worked with bees because he really loved bees and tried to study their genetics. But again, the other monks that were using the gardens and outdoor areas were extremely irritated because I guess these were particularly aggressive bees that he worked with. And then he did eventually settle on working with pea plants, which were benign and pretty to look at. Nobody complained about those. So let's look at some situations that we call extensions to Mendel. They still follow Mendelian genetics, but there is something additionally going on that makes it look different. Okay. Polygenic inheritance. It occurs when multiple genes are involved in controlling the phenotype of a trait. Multiple genes. So not just one gene on one pair of homologous chromosomes, but multiple genes, usually across many of the chromosomes. And the phenotype, remember, the phenotype is the physical representation of the trait. It is, in these cases, an accumulation of contributions by all of the genes that contribute. These traits are often continuous and are referred to as quantitative traits. Okay, so what does it mean with an example? Let's look at human height. Okay. You know that unlike pea plant flower color, purple or white, which is just totally binary for the species that Mendel chose, in humans, the height is not binary. There isn't tall and short, right? We have heights of all variations. And if we were to line up people by height, we would see something like this. This is actually, I think, Harvard class of like 1920 or something, lined up by height. You see that it produces more or less a normal distribution of bell curve where most people have a height of somewhere in the middle, right? So the height of the bar is the number of individuals, and on the x-axis we have height in imperial system. So there are a lot of people that are somewhere between 5'5" and 5'11", but there are a few people that are around 5 feet and a few people that are around 6 feet tall. That's because in order to be super tall, you would have to have inherited all the tall causing genes from both of your parents, right? And what causes you to have a certain height? And that has to do with the length of the long bones, so the leg bones in your body, and the spacing of your spine bones, and how thick the discs between your spine bones are, and how long your neck is, and how oval your head is. And it also has to do with the density of your bones and parts about development when you were a child, of how those bones grew and how you were able to absorb calcium, all kinds of genes that lead you to be of certain height. And if you inherit only the ones that happen to add to being of tall height, then you will be particularly tall. It's also less likely that you'll be particularly short, because you would have to inherit all the alleles that cause your height to be shorter. It is much more likely that you inherit some combination of alleles that make you taller and some combination of alleles that make you shorter. So you become some sort of medium height. The same goes for skin coloration. If you have two medium colored individuals, then it is likely that the offspring will inherit some of the darker causing alleles and some of their lighter causing alleles. So you'll be somewhere in the middle. And only very few individuals, like 1 out of 64 and maybe this 6 out of 64. So these 7 out of 64 that are the particularly dark skinned, and these 7 out of 64 over here that happen to inherit most of only the light and are pasty white individuals. It's a lot more options for combinations. So skin coloration, of course, is distributed across the spectrum, and if your parents are medium color, then you're likely to be medium color as well. However, if both your parents are medium color, it is possible that you can be really, really light, way lighter than both of your parents, or you can be much, much darker than both of your parents, because there are many genes involved. In the previous diagram, they only mentioned 3 genes that are responsible for human skin pigmentation. In reality, there are more genes that are responsible, so it's even more complicated. I would never ask you to do a Punnett square with 3 traits. We have computers that do that now. It's very, very tedious and takes a long time. All right, so polygenic inheritance. Many genes are responsible for one phenotype. And because of the combination of that, you can have a whole spectrum of phenotypes. Okay. Moving on with extensions to Mendel is a concept called pleiotropy. It refers to a situation when an allele, which has more than one effect on the phenotype. It's almost a little bit like the opposite of the polygenic inheritance. Pleiotropy effects are difficult to predict because a gene that affects one trait often performs some other unknown function. And this is very common. Especially it's common for phenotypes of different metabolic functions. So when we're talking about proteins that don't cause some sort of outwardly appearance like color of hair or skin or eyes or your height, but it has something to do with like how well something is metabolized inside of your body. Okay. So one example, it's the classic example, this is for diseases like cystic fibrosis or sickle cell anemia. And we will look at the sickle cell anemia. It is the most common genetic disorder for people of African descent. And what ends up happening is that people who have a sickle cell allele, so this is just have to get a little bit more into it in order to explain this to you. But here we see red blood cells, and they are in a very weird shape. We actually call it kind of sickled shape. In reality, fully functional red blood cells should just be tiny discs with a little indentation at the front, kind of like what are those candies? Wow, now I can't think of it. Okay, so the reason why these cells look the way that they do is because this person inherited alleles that cause the hemoglobin, which is a protein inside of your red blood cells that carries oxygen. And we have many, many, many copies of the hemoglobin inside of each cell. The hemoglobin doesn't have the right shape, and so the many numerous hemoglobin proteins inside of a single cell, they stick to one another in like a rod shape. Instead of just being round and floating inside of that cell, they are sticky to one another, and they form these rods, which elongates a cell. Okay? So someone inherited an allele for the sticky shape of hemoglobin from mom, and they also inherited an allele for this messed up hemoglobin shape from their dad. So they only produce hemoglobin protein that is this deformed shape. This causes some immediate consequences, which is the breakdown of red blood cells, clumping of cells, and clogging of small vessels, because the cells themselves, even the cells themselves, are not round, and therefore they don't pass through the really tiny blood vessels the way they should. They get stuck and back up the blood, right? You can also get accumulation of sickled cell in the spleen, and this causes spleen damage, damage to other organs, brain damage, pain and fever, heart failure, anemia, physical weakness, impaired mental function, paralysis, pneumonia, and other infections, rheumatism, kidney failure. All of this can be caused by a single gene, which is hemoglobin shape gene. And there are many other consequences. Luckily, in the last 2 or 3 decades, sickle cell anemia can be treated relatively well. So people have to go to the hospital and get some transfusions and other interventions, but people can live pretty, healthy lives without mental impairment and paralysis and all of these really harsh consequences up until well into adulthood, whereas just a generation or so ago, people died in childhood. Sometimes just as when they were babies, because of such huge consequences to this one gene. Okay, so pleiotropy is one gene, and even more specifically, a specific allele of that gene. So in my example, the incorrect recipe for how to fold hemoglobin causes many effects. Okay. Moving on to a different extension to Mendel, and that is that there can be more than 2 alleles for a particular gene in a population. For example, let's look at blood type. I don't know how many of you know your blood type. I only found out my blood type, I think, when I was pregnant, or if you donate blood, you will also find out what your blood type is. Blood type is based on several different things, and in this case, we're only going to look at the very basics. And what it's talking about when we mean blood type is what shape of carbohydrates you have attached to the phospholipids on your red blood cells. So these little red discs are your red blood cells. This particular individual has one kind of carbohydrate. We will just call it carbohydrate A expressed, or it's stuck. Remember when we learned about cell surface markers back in a chapter where we were learning about membranes? Well, it's one shape of carbohydrate. Some people have a different shape of carbohydrate. We'll call it carbohydrate B. It's also possible to express both of the carbohydrates, and it's possible to not have any of those carbohydrates on the cell surface. So let's say there are 3 of the alleles: carbohydrate A, carbohydrate B, and no carbohydrate expressed at all. That's 3 alleles for this particular trait. Each individual only has 2 chromosomes, right? We have 2 homologous chromosomes for each type of chromosome. On each of these homologous chromosomes, we can carry one allele or one version of a gene. We cannot carry all 3. So I could have in me the instructions for how to make A and B, or B and O, or A and O, but I cannot carry instructions for how to make all three of these options. Okay. So if Mendel was working with traits that had more than one allele, he would not get the very nice and exact ratios that he came up with. And it would have been very difficult for him to come to the conclusions that he did, because the data would have been all over the place. Next, for some extensions to Mendel, is incomplete dominance. Incomplete dominance kind of makes it look like the people who were talking about traits blending were right, but spoiler, they were not right. This is when you have true breeding red flower plants, and this is not talking about peas anymore. This is only happens in a few species of flowering plants. Okay, you cross a red flower with a true breeding white flower of the same species, and your F1 generation has an intermediate phenotype. They are all pink. So looking here, this is dragon lily, and for this particular case, when you cross these two true breeding plants, you get an in-between phenotype. So the heterozygotes are really easy to tell apart because they look different than either homozygous dominant or the homozygous recessive. It is really difficult to explain in a short amount of time how this can happen. But the reason that this is happening is because in this particular case, not in all flowers, definitely not in the peas, in this particular case, in order to make the molecule that causes this redness, there are actually several steps. And when you have two alleles for the color making, you're able to complete the whole process of the synthesis of those different steps so that you have full pigment. If you're heterozygote, you can only complete part of the synthesis of the pigment, and so you do not have enough information. It may be frustrating to just accept it as is, but if you are super interested in this, please continue on your studies in genetics and biochemistry, so then it becomes a lot more clear. If you have particular specific questions to this, please ask. Another extension to Mendel is called codominance, and that is when the heterozygous is not a in-between phenotype, but they express both. The traits are not, they do not look like they're blended. It looks like totally both are expressed. An example of this is the AB blood type. I already mentioned how the ABO blood typing represents multiple alleles. It also represents codominance. So let's look at what I mean by that. If a person inherits type A blood type from their mom, and they inherit type B blood type from their dad, they are actually inheriting two variations of how to produce a carbohydrate on the surface of the blood. And they end up expressing both the A and the B. They're both dominant. They're both two versions of a dominant allele for this gene. The recessive allele for this gene is just not making any. So an individual with either type of the dominant alleles will cover or overpower the recessive allele. But if an individual inherits both of these dominant alleles, they will represent them both equally. So that person will have AB blood type, and they will be expressing both of the carbohydrates. This is an individual that can theoretically get blood transfusion from any of the other blood types because their immune system recognizes both of the carbohydrate shapes as self, right? This is how we spot intruders in our blood and lymph, is by white blood cells feeling around the outside shape of everything that they touch. And if they see either of these carbohydrate shape, they're like, oh yeah, that's our own body, that's fine. However, if someone is type O blood, they cannot receive blood from any of these other individuals because their immune system wants there to not be either of these carbohydrates on their surface. However, type O blood people can donate blood to A, B, and AB people. In reality, there are other types of markers. You've probably heard about being like A positive or negative. The positive and negative is the Rh factor, and it's a different type of labeling on blood cells. It's an additional type of labeling on blood cells that is coded by a totally different gene, which interacts totally different and separately and has nothing to do with this ABO system. It's just that the ABO system, as well as plus minus Rh factor system, are the two most important types of markers on blood cells. So we combine information to say, hey, I'm B positive, right? Interestingly, depending on what part of the world you're from, you're more likely to be a certain blood type. So in some places, O is very rare, or AB is very rare, or B is rare, or A, while in other places that distribution is very different. Of course, we're such a melting pot here in Portland that it's likely that we have pretty equal distribution of these traits. Okay, so blood typing is a representation of two features, and that is codominance as well as multiple alleles. Moving on, environment, of course, has an influence. And you can think of, let's say there are two twins that are identical to each other, so they have the same DNA, but one of the girls uses tanning beds on a weekly basis, and the other one lives in a basement and plays video games and never sees the sun. Over a period of a few years, they would look different. They would have different phenotype for their skin color, right? Or someone who is maybe malnourished or isn't able to get enough of a certain vitamin, that will affect their phenotype for height or certain development features, right? So it's not all about your DNA. However, here's an example of a coat color in Siamese cats, and also this is true for Himalayan rabbits, that demonstrates the environmental influence very, very directly. So there is an allele that produces an enzyme that allows pigment production only at temperature below 33 degrees Celsius. So what this means is that at a certain point in the development of the cat or the rabbit, right, when they're really, really little, they if there is a body part that is cooler than 33 degrees Celsius, then a pigment will be produced in that part. But parts that are higher than 33 degrees Celsius in temperature will not produce the pigment. So even if you have the allele for making this brown color, you will not see it produced unless you have these temperature differences. Okay, so environment can influence on our phenotype. It's another layer on the genetics that's extremely complicated. Due to time, I'm actually going to skip epistasis because it's quite complex and takes a long time to explain, and I think we have enough examples. Here is a slide of summary of the different relationships. So complete dominance, which is what was described in Mendelian genetics, have incomplete dominance, codominance, multiple alleles, pleiotropy, and polygenic inheritance. You can forget epistasis for our class this time. Make sure you know the names of these relationships and you understand how they would be expressed. So if I give you an example, like a green Martian has a baby with a yellow Martian, and all of their offspring are spotted yellow and green Martians. What kind of inheritance is that demonstrating? Well, both phenotypes are expressed equally. Without blending, that would be codominance, right? There's one more difficulty to understanding genetics, and we will use human examples here. And this is traits that are physically located on the X chromosome. These traits are inherited differently in men and women. And the reason for that is because women have two X chromosomes. So for biologically female humans who have two X chromosomes, these chromosomes behave just like any other chromosome. You have two homologous chromosomes. They each have one allele for the same gene. So you end up having two alleles for the same gene. So in individuals that have two X chromosomes, like most females, there isn't anything odd about the inheritance from the first look of things. This is a caveat I'm going to come back to. Males only have one X chromosome. They also have a Y chromosome, but a Y chromosome is not homologous to the X. I mean, look at it. It's not the same shape at all. It's not the same size, and it does not carry the same genes. In humans, when embryonic development begins, the human fetus develops as a female unless at a certain point of the development, information on the Y chromosome, especially from the SRY region, starts to produce a protein that derails fetal development from female to male. If Y chromosome is absent, then of course that protein is not going to be produced and is not going to change the development into male. Or actually, a person can have Y chromosome, but the SRY gene or SIY region on the Y chromosome may not have the correct genetic information, or that section of the chromosome can actually be broken sometimes, and it will still not produce the product, the protein that would then direct development to change from female to male. Okay. So here is a shortened map for the Y chromosome. It has only a few genes, and none of them are absolutely crucial for survival. Obviously, most females don't have a Y chromosome, and therefore they don't, they can still be alive. You don't need to have it. So the way the inheritance of the sex chromosome happens is remember, we have a total of 46 chromosomes. That is 44 of the autosomic chromosomes or autosomes and 2 sex chromosomes. Your father will generally have, in kind of basic situations, will have one X and one Y. So when he makes his sperm, 50% of the sperm will have the Y and 50%, more or less, will have the X chromosome. Of course, the other chromosomes are also segregating all and mixing up, right? Whereas all female eggs have the 22 autosomal chromosomes, and all of them will have one X chromosome if meiosis happens correctly. Therefore, depending on whether this egg combines with this kind of sperm or this kind of sperm, you will get a biologically female zygote and a biologically male zygote. As you may imagine, this has a consequence of how the traits that are found on the X chromosome are expressed in biological females, because they have two versions, they have two alleles, versus biological males, which have only one allele on the X, and they do not have a homologous chromosome there. So there's only one copy of all the recipes on the X. We call these sex-linked traits. There are also sex-linked diseases. Some examples are Duchenne muscular dystrophy, which is a common disease where boys have muscles that develop normal, and then at some point in their childhood, they begin to deteriorate really quickly, all the skeletal muscles. Hemophilia is also a disease that affects males much more frequently. It causes people to not have the protein required for blood to clot. So people bleed out. This used to be called royal disease. I'll show you why in a second. And color blindness of a particular type of color blindness is a sex-linked disease. Okay, let's look at how we would do a Punnett square for any trait that is located on the X chromosome. Okay, the way we write it is we have to specify that we're talking about the X chromosome, and the big or little letters, remember how with Mendel's P's we had big P and little p, we still use those, but they are superscripts over the X chromosomes. The Y chromosome is not relevant, but we include it without a superscript because remember, it doesn't carry the same genes, so it doesn't have the N that we're talking about. But we include it in the Punnett square because it gets inherited together with these, and it's to remind us that the offspring will be male. Okay, so this woman who is homozygous dominant for N, and this male who is, we call it hemizygous, it's not homo or hetero because there's only one, but he's recessive for this trait, right? When he makes sperm, some of the sperm will have the X chromosome with the recessive trait N, and some sperm will have the Y chromosome and they will not carry at all the trait for N. The eggs will all have X chromosomes that carry the dominant trait allele for N, right? Because that's the only thing that's possible in the eggs. When you cross it, let's see what we got. Here we got a female, because XX makes a female, biologically speaking, heterozygous female. Here we have a heterozygous female. Here we have a dominant hemizygous male. And here we have a dominant hemizygous male. Okay. So if having this little n causes someone to have disease like hemophilia, would any of these offsprings have hemophilia? No, because it's a recessive disorder, so it would require you to have two little n's. Think of it like the white flower color for Mendel's pea plants. The only way to be white is to have two of the recessive alleles for color, or two little p's. In this case, if this is not flower color white, but this is having hemophilia, or the problematic protein for blood clotting, or actually an absent protein for blood clotting, none of the offspring would have that phenotype. None of them would have that disease. So this match, this female and this male, would not produce any offspring. There's 0% chance they would produce offspring that have hemophilia. However, if an individual that is heterozygous, I should go here, a female that is heterozygous for this trait, mates with a male who has hemophilia, then 50% of the males will have hemophilia. Okay, pause it and see if you can re-say it in your own words. Also, if this heterozygous female has offspring with this totally healthy, non-ill male, notice that 50% of the males will have hemophilia. 50% of the females will be heterozygous, or they will be carriers for the hemophilia allele. Whereas here, 25% of the 25% of the kids, but 50% of the males will have hemophilia. 50% of the females will have hemophilia. It's very unlikely because men who are hemophiliacs are generally unlikely to reproduce. So hemophilia is called a royal disease because it seems like the Queen Victoria of England had one allele for hemophilia. So that means she was heterozygous for hemophilia, which is a trait found on the X chromosome. Because she was biologically female, she had one X chromosome that produced a normal blood clotting protein and one X chromosome that did not produce a normal blood clotting protein, but you just need to have some in order to have normal vascular system. So she obviously did not know this. And in that time, all the royals in Europe interbred like crazy, and there was also a lot of incest happening. So if we look here, this is a royal pedigree. Each circle represents a female, each square represents a male. The line between them represents them having a sexual union that produced offspring, which are on this bottom row. If the box or circle is colored in, it means that they were affected by the trait that's in question. So in this case, we're only talking about hemophilia. You can see how Queen Victoria, she had a normal phenotype, did not have a disease, but she was a carrier for the disease. She was heterozygous on the X chromosomes for the hemophilia causing gene. She passed that on to two of her daughters, and she had lots of kids. Like, that's a love story. The Prince Albert and Queen Victoria, that's quite a love story. And one of their sons, Leopold, was actually hemophiliac. He did have offspring, and his daughter was a carrier for hemophilia. When she had a son, he had the disease. But notice how it got spread through the Prussian royal house. So this is what we would now call German, more or less. Russian royal house. And this is actually the whole saga. Why, for big reason for why the Russian Revolution happened and why communists like Lenin and Stalin came to power, and that whole overthrow of like European history that certainly very much affected my family's life. It was all because of the hemophilia that Tsar Nicholas's son Alexis had. They only had one son. They were very focused on him becoming the heir, for the next generation. And he was a hemophiliac, so he's constantly sick, and that caused just all these problems and abandonment of his Tsar duties. Anyway, this is why the Bolsheviks executed the whole family, all these children, including Anastasia, yes, the cartoon, great cartoon, totally historically inaccurate, but great, as well as Alexandra, who was Queen Victoria's granddaughter, and Tsar Nicholas. They were all murdered, and that's horrible. But there's just very complicated history that was very much affected by the fact that Alexei, their only son, had hemophilia, which was passed on genetically from the great-grandma Queen Victoria. Also, the Spanish royal house got affected by it, and you can see all the shaded-in men who died from it. Okay, so males get affected by traits that are expressed on the X chromosome, much more than females, because you don't get two options, like two recipes. Oh, if one recipe doesn't work, then we can read the second recipe we have and hope that that one produces what we need for the body to function. That's what females have, because they are diploid for the X chromosome, while males only have one. However, the story for females is not so simple. Something happens called dosage compensation. And this is very interesting. It is that when you, as a female, biological female with two X chromosomes, when you develop during embryonic development, you read information on both X chromosomes. However, at some point in the fetal development, each cell, of course, each cell has two of the X chromosomes, it will shut down one of the X chromosomes. And from there on out, only information on the non-shutdown X chromosome will be read. Every future generation of cells that comes from that cell that has the X shut down will produce cells that also have the same X chromosome shut down. So in this way, females, when they come to even to be born, and of course when they're adults, they are actually only reading one X chromosome and ignoring the other. However, because at the beginning of their fetal development, they were reading both, it's like all of their tissues are patchy for both of the versions of the X chromosome, which compensates for some of the problems that males experience with genetic disorders of the X chromosome. The deactivated X chromosome is called a Barr body, B-A-R-R, after the scientist who described them first. And therefore, females are genetic mosaics. They are patchy for information expressed on both of the X chromosomes. And this can be demonstrated by a calico cat. If you've ever had a calico cat, it is very likely that it was a female. And the way that their fur color is expressed is that on their X chromosome, they have alleles for black or red pigment color, or I guess I should say orange pigment color. And at some point in their fetal development, there was a cell here that deactivated the orange fur color, and all of the cells that on the skin surface reproduced by mitosis from that point on, they also all expressed the black color. Whereas over here, it was the X chromosome that carried the black colored allele that got deactivated. And so all of the cells that became the future generations that grew in this area from that original cell now also express the orange color. Under a microscope, that Barr body is actually stuck to like the surface of the nucleus, and it's all like super condensed and is not read for any information. Okay, it just gets less condensed for mitosis, so it can be replicated and passed on to the next generation of cells, but it's not used for actual cell maintenance. In humans, we have disorders. Most of our genetic traits and disorders are inherited recessively, and that means that in order to express what we would call the disorder or the disease or this particular trait, the individual has to be homozygous recessive, like the white flower color. And just like in the white flower color for the pea plants, it's usually because a protein is actually not being created. So if you don't have another copy, like here, a dominant second allele that does have the correct recipe to produce something, a protein, information for how to make a protein, then which can kind of compensate for the other allele, then you will have the phenotype that is that recessive homozygous phenotype. So here are these super adorable siblings, sister and a little sister or brother, or maybe, yeah. And this little brother or sister are albino, and that means that they do not produce melanin that gets deposited into their skin or hair, right? So they're missing both options for recipes for how to make melanin, the protein that causes pigment. Heterozygous individuals have normal phenotype because they do have one allele, one version of the recipe that produces melanin, and that's usually enough. Depending on the trait, it can look exactly like the phenotype of a homozygous dominant individual, but they are a carrier. So if a heterozygous individual has offspring with another heterozygous individual, then of course 25% of their offspring have a chance of having the recessive homozygous phenotype, in this case albinism, but it can also refer to Tay-Sachs disease, cystic fibrosis, and sickle cell disease, which we've already talked about. This is why in communities that interbreed, where cousins marry and have children with other cousins, you're more likely to have some of these things pop up. We are all carriers for stuff, but the less related you are to someone, the less likely you're carrier, you're heterozygote for the same types of alleles. So it is less likely that you'll produce some kind of problematic or health, difficult phenotype. Here's an example of how sickle cell alleles are working. The two homologous chromosomes have this area that codes for how to make hemoglobin. And in this case, the hemoglobin from both recipes is red, but both recipes are it's like they have mistakes in them. That DNA code has a mistake in it. And when you read it and you make a protein exactly based on that code, just like a recipe, you produce a product that's not quite right. And the hemoglobin has this sticky shape. See how they're notchy. And when you have many, many hemoglobin particles inside of a red blood cell, they clump together to create these rods, which sickles the blood cells. In an individual that has both versions, how to make the regular protein hemoglobin and the sickled, they generally destroy the blood cells that happen to have a lot of the sickled or the problematic hemoglobin really fast, and you make more new red blood cells with the hope of having more of the correct protein. And generally that individual does not have a lot of health issues. Most of them do not even know that they are heterozygous. So having one recipe or one allele that has code for the correct protein is super helpful. However, there are certain traits that are inherited dominantly, right? So this is when, instead of like here or in the hemophilia situation or in the white flower color situation, it's just a lack of properly functional protein that causes the phenotype. In dominantly inherited traits or dominantly inherited disorders, so those are traits that we would as humans define as disorders, it is actually the making of something additional problematic instead of the lack of something functional. So in these cases, if you have any of the dominant alleles, you will express the trait that we're talking about. So one type of genetic dwarfism, which is called achondroplasia, is a dominantly inherited disorder. So these are more rare, but if an individual in this situation has any dominant alleles, so in this case, if you're even one dominant and one recessive, the dominant allele is the one that's, it's the one that's causing the problem. And so having it makes individual express that phenotype. So that means if you're heterozygous for that trait, you will be achondroplasia. You will have achondroplasia. This is actually a semi-famous surgeon who had achondroplasia himself, and he was a expert on surgeries to help people with achondroplasia, and part of that group of individuals who were doing research to make life better, because it's not just about a short stature. There are also other health problems that come from being achondroplasic. Okay, so the only way to not have achondroplasia is to be homozygous recessive. Okay, so these are dominantly inherited disorders where you have at least one of the dominant alleles, you will express that trait or that have that disorder. Recessively inherited traits or recessively inherited disorders are ones where you have to have two of the recessive alleles, and that's the only way to express that trait. Okay. Many human disorders are actually multifactorial, and that means that there are many genes as well as environmental factors that cause the particular type of disorder. Heart disease, diabetes, cancer, alcoholism, and all types of mental health illness have this kind of complex combination of genetic and environmental components. At one point, I did a lot of research on the genetic components of type 1 diabetes. And at the time, there were over 50 different genes that are attributed to contributing to someone being prone to type 1 diabetes. It's like you have to inherit a particular cocktail of all those different types of alleles. And then you have to have a particular environmental pressure or something happen, usually a viral infection at some point in your childhood. So only if you have that full cocktail of just specific combination of all those genes and there is something happening to your body, something environmental, that you are likely then to have type 1 diabetes. It's extremely complicated to study multifactorial disorders, and the research is ongoing for all of these listed and many more. Okay, that's it for our basics of genetics. Please make sure you pause, go back, study your textbook, retell the stories, as I say, and take notes in order to internalize this information. See you next time.